Hello and Bujeri Kamaru, which means hello in the language of the Gadigal. I acknowledge the 29 clans of the Eora Nation, the traditional owners of the land now called Sydney, where I am based. I pay my respects to elders past and present and the essential living relationship between all First Nations peoples and the land, water, and the ocean. Let us know where you're dialing in from today in the chat function, which you should find in the box on the right-hand side. There's also a question box beside the chat function, and we encourage you to submit your questions throughout the webinar. And we hope to have at least 10 to 15 minutes at the end of the talk for a great Q&A session with our fabulous speakers today. So welcome to another edition of ALA's Science Week webinars. This one is focused on citizen science metadata. My name is Erin, and I'm joined here by my co-host, Pam Slatyer. We're one of several staff who work for the ALA across citizen science. So in this webinar, we'll hear from both Jasmin and Thomas about what makes a great species observation record and learn tips and tricks for collecting the best quality metadata. But before I kick off, I just wanted to share just a few slides articulating why citizen science is so important for the ALA. So this first figure is from a paper that's recently been accepted, but not yet published. So you're getting a bit of a sneak peek here, where we broke down the data in the atlas between citizen science and non-citizen science. So by non-citizen science, it's essentially everything else. It's data we receive from industry, research, museums, and collections, for example. And although both data sources are growing, which is fantastic, you can see that citizen science has really taken off, particularly in the last few years. These are some stats generated by CAM, and I think they really show how important citizen science has become for the Atlas. So over the past year, we've added about 10 million observation records to the Atlas, and 95% of these were citizen science. Within this 95%, 75% were bird data, 18% were from iNaturalist, so all taxa, and about 1.3% were from other sources. And excluding birds, there are just over 1 million observations in total nationally. So the next 3 slides, I just wanted to sort of explore these bullet points a little bit more. So again, this is from the paper that hasn't yet been published, but we're looking into the data in the ALA just a little bit more. And Dax Kelly, one of our co-authors, actually produced these figures. So this first figure is of the shingleback skink. And again, we've broken down the observational records between citizen science and non-citizen science. And as you can see here, there's actually a lot more observations from other sources. This is okay though. I think it really highlights the importance of the complementary nature of data in the ALA and the value of a large data aggregator. But we also think it might be because, to the best of our knowledge, there isn't a large citizen science project encouraging observations on the shingleback. But if we turn now to look at the distribution of the parentsfree frog, I think many of us will be familiar with some very large citizen science projects operating in Australia that are encouraging observations of all frog species. The picture changes a little bit, and we can actually see that about 1/3 or 33% of the hexagons are contributed by citizen science in this example. In this final slide, looking at the red-browed firetail, the observational records are heavily dominated by citizen science. In this case, I think it's 84% of hexagons. And of course, we've had in Australia really long-standing citizen science projects focused on birds and an avid birding community. And so I think this is really reflected in this figure. So I guess reflecting on this a little bit more broadly and what this means for the Atlas, given citizen science is so important for us, we have to consider things about how we get more out of citizen science. Data quality, which is what we'll be touching on today, is certainly one aspect, but also things like taxonomic bias and coverage. And this figure is actually taken from an article looking at GBIF data and the biases within that. And the ALA largely reflects and has similar patterns. Just finally, though, I did want to reflect on a good news, good impact story, the biosecurity alert. So a few years ago, the Atlas partnered with state and territory and federal agencies and the biosecurity alert system. So essentially, these government agencies provide us with a list of species that they're interested in. These species are largely based on the EPO list or exotic environmental pest list, but not always. And when a species that matches the list comes into the atlas, we then ping these users with an email alert. And what's interesting here is that 99% of the observations that we're then passing on to management are coming from citizen science. And I think this small example really demonstrates the value of citizen science with a biosecurity lens. So in terms, and just to conclude, what's next for the Atlas in terms of supporting citizen science? Well, we want to turn our attention to how we can direct national citizen science efforts to operate in a framework around gap filling. We want to assist with guidelines on how to collect higher quality data, and this webinar today is the first step, essentially, and really try and provide more structure around citizen science contribution. So with that, I will stop sharing. And I will pass it over into the very capable hands of our first speaker, Thomas Mesaglio, who is a PhD candidate at the University of New South Wales. Thomas is currently researching plants, but for those of you that know Thomas, he has his fingers in many pies. And he's also worked on fire recovery, marine forensic, and invertebrate taxonomy. Thomas is hugely passionate about biodiversity and citizen science and the intersection between the two. And I think to reflect this, he's a forum moderator and curator on iNaturalist, and Thomas regularly organizes and leads bioblitzes around Australia. So over to you, Thomas. Thank you so much.
Awesome. Thanks a lot, Erin. So start sharing. Okay, so I'm largely going to delve into marine invertebrates, some terrestrial arthropods, and plants today. But before I get into the thick of things, just wanted to really emphasize something that Erin already mentioned, and that's that the ALA is a data aggregator, and in that sense, it's not only taking citizen science records just from iNaturalist, which probably dominates a lot of these statistics, but also from a wide range of other citizen science platforms as well, not just limited to those I've got on the screen here as well. So I guess the point I'm trying to make here is that it doesn't matter how you're contributing to citizen science or what taxa you're interested in, all of the platforms are equally important, and it's fantastic to see contributions from all of them. And also the tips and tricks that I'm going to talk about and that Jasmin is going to talk about really apply to a lot of these platforms as well. So it doesn't matter how you're contributing, all of the content today is really relevant. Now, for any given citizen science record, there are 1,001 different kinds of metadata that you can possibly extract from them. But largely, no matter what kind of data point you have, you can really pull out three main types of metadata that are the most important ones, no matter what taxon and no matter where you are. So the first of these is a spatial element to the metadata. So this can be in the form of written place names, but really the most important piece of metadata here is the actual coordinates themselves, so the latitude and longitude. Then for the vast majority of citizen science records, you have some kind of evidence to support the sighting of that organism. Now, of course, in many cases, that evidence is going to be a photograph or a set of photographs, but you also have sound recordings as well. So frog ID is something many people would be familiar with from that perspective. And then finally, you also have some kind of date attached to that record. Now, given in 99.9% of the time, no matter what you're recording your citizen science data with, whether it's a tablet, a phone, a camera, that date is almost always going to be auto-populated. So I won't delve into the date here. But what is important is the spatial data. Now, if you are taking photographs or sound recordings with your phone, and assuming you have location services turned on, then again, 99% of the time, that location, those coordinates will be auto-populated for you. But even though camera technology has advanced massively in the last 5 to 10 years, it seems to be the case that for most DSLR cameras, they still won't automatically geotag your photos for you. So in those cases, if you're out and about, you're on a big bushwalk, you've taken lots of photos with your camera, you get back home, you're actually forced to manually input the location for all of those records. Now, not only is this obviously massively time consuming on your part, but it also means that that metadata isn't as accurate as it can be. You know, if you've been in a national park and you've been walking all day, you've taken hundreds of photos, chances are you aren't going to remember exactly where you saw each of those organisms. So what there is nowadays in the market is a fantastic range of very cheap and very easy to use geotagging apps. So the one that I use in particular is Geotag Photos Pro 2, costs around $15. You download it onto your phone, and then you also download it onto your computer. And all you have to do is ensure that the time on your phone and the time on your camera are synced, and you just have it running in the background the whole time you're out walking. You get home, literally press a single button, and it will geotag hundreds or thousands of photographs for you. So a huge time saver, and also ensures that that particular metadata is as accurate as it can be. Now, as I mentioned, the core of citizen science data does tend to be photograph based, but we can also have many instances, Frog ID being one of them, where sound recordings are perfectly valid as well. And in fact, for many groups of organisms, not just frogs, it is indeed sounds which are the most reliable way of identifying those organisms. And I think a classic non-frog example of that is cicadas. So yes, there are some classic charismatic cicada species, things like green grocers or double drummers, that are very easy to identify from photographs. But there are also many smaller genera, things like Yoyeta or Porosolta, where you've got lots and lots of really, really similar, if not almost identical species from a morphological perspective. But if you record their calls, they're all really unique, and it allows you to identify them a lot easier. But the problem often is that when you're recording sound with your phone or your camera, it will generate a video file, whereas platforms like iNaturalist only accept audio files. But thankfully, once again, we've got really easy to use software. And in this case, it's actually one better than the geotagging software, in that most audio converters are completely free. So no cost involved. So the particular one, again, that I use is Online Audio Converter. You upload your video file, convert it to an M4A, which is a type of sound file, and then you can upload that exactly as you would any photograph. But to really get into the thicker things now, I will delve into the world of photographs and ensuring how you can take the best photographs possible across a wide range of taxa to really maximize the identifiability of those particular organisms and ensure that that particular type of metadata is as robust as it can possibly be. So for anyone who's been out and about and beachcombed before, I'm pretty sure that you'll have seen at least one cuttlebone in your life. So these are the internal shells of cuttlefish that they control the buoyancy of the cuttlefish. And they're a fairly common sighting that found washed up on the beach because they actually float on the surface of the water. Now, most of the time when I do see people uploading photographs of cuttlebones, they tend to take just one photo, and that's from a dorsal perspective. So that's the top cuttlebone that you see in each of these 4 plates. But actually, one of the more useful angles that you can actually take is from underneath the cuttlebone, so the ventral view. In particular, so you can capture these 2 structures that I've marked, which is the inner cone and the outer cone. So if you do find a cuttlebone washed up, all you've got to do is take a photo of the top side, flip it over, get a photo of the underside as well, and you don't need to ever have seen the actual cuttlefish itself. More often than not, you will be able to identify your cuttlebone species as long as you've got these 2 different photos. Now, the exact same concept applies to bivalves. So these are shells that have 2 parts to the shell, so things like oysters or clams or mussels. Again, you want a photo both from above and from underneath. Now, in this case, the photo from underneath might not necessarily get you a species ID, but it's really important for coarser level IDs to taxonomic levels like family, where the shape and the size of the hinge and the hinge teeth, so this is where the two shells join, can be really important for those coarser level IDs. And just to hammer home the point with one more example, same thing applies with gastropods as well. So particularly if you have these kind of tall, relatively skinny sea snails, you want to get a photo from underneath so that you can see the aperture here where the snail comes out, and then just rotate that shell 180 degrees and get a photo of the other side as well. However, if you find the more snail-like gastropods that have a more globular shell, ideally you want to get a third angle as well. So in this case, you get a photo from directly above, flip it 180 degrees, get a photo of directly below, but then also slip in this extra shot, which corresponds with one of the ones on the previous slide. And once you have these three shots, again, 90% plus of the time, you'll be getting a reliable species ID. But I think really from a beach combing perspective, and particularly seashells, the number one piece of advice I can give is to actually take photographs of the shells in your hand rather than just leaving them on the sand. Because what this does is it immediately provides another type of metadata, which is size. Now, of course, it isn't going to give you an absolute measure of size down to the exact millimeter, but relative size is really, really important for many shells. And in fact, there are a lot of examples where you may have two quite similar shells, where one is, say, as big as your hand, whereas another one might only span a couple of finger widths. So if you hold that shell in your hand, it immediately gives someone who is looking at that record a sense of the relative size of the shell and is really useful information for then pinpointing an ID. Now, things get a bit trickier when we dive into the world of insects and other terrestrial arthropods, just because there is such incredible diversity. So more often than not, when you go from order to order or family to family or even genus to genus, you might have to take a radically different set of photographs in order to maximize identifiability. So largely the main piece of advice I can give when you are dealing with insects and most other invertebrates is just try and get as many photographs as you possibly can and really hedge your bets. And even if you had no idea what photo you actually needed, if you take as many as you possibly can, well, chances are you'll probably end up including that angle. But for a few specific groups where people are more likely to encounter, I can give some more specific tips. So here I will give a quick shout out to the Christmas beetle count, a really fantastic project run through University of Sydney and Invertebrates Australia. You just upload your records of Christmas beetles to iNaturalist and they automatically funnel into the project. Most times when I see people uploading photographs of Christmas beetles, they tend to be a photo that looks something like one of the two on the left-hand side here. So either from the side or from directly above. And whilst these are useful to include, for many Christmas beetle species, it's actually a photo of the back side, which can be the most useful shot for identification. So things like the shape, the size, the color, and the hairiness of the backside are all really useful features for getting that photo ID to species. Now, kind of a similar concept to our seashells and cuttlebones with spiders. If you do see a spider in a web, so this will particularly apply to orb weavers, not only is it good to get a photo of it from directly above, so the dorsal surface, but if you're able to walk around to the other side of the web and get a photo from underneath, that is also tremendously useful in many cases. And if we're dealing with flies, well, the number one feature that I can recommend is actually the wing venation. So the pattern of veins in the wings. So even if you're unable to get any other shots, or if some of the other areas of the fly are maybe a little bit out of focus, if you can get those wings in really great focus, then it is a hugely beneficial feature to have in your photographs for fly ID. And this really interestingly actually ranges across all taxonomic levels within flies. So the wing venation is actually one of the most important features for identifying flies to family. But as you can see in this specific example here, where Chris Lampkin, who's a massive specialist in Australia, the top expert in bee flies and a few other fly groups, she not only provided an identification to genus for me, but actually even to species based purely on the pattern of veins in this fly's wings. So certainly this is the most useful shot. If you're only going to be able to get one photo of a fly, which often ends up being the case given how skittish they are, and they can be pretty difficult to get close to. So if you're just planning to get one shot before it flies away, this is the shot that I would recommend if possible. Now diving into the world of plants, a quick transition. Plants are pretty similar to insects in that they have pretty high species diversity, not quite as high, but nonetheless still high. And that as you go from family to family, again, genus to genus, the types of photo that you need to take are often pretty idiosyncratic and will often vary quite dramatically. But thankfully, plants have this one big benefit in comparison to many insects, and that's that they don't move. So yes, you can go in with good intentions when you're photographing insects and say, I'm going to get photos from the top, the side, of the face, of the backside. But then when you actually get there and you take one photo and the insect flies off and you never see it again, those plans often go awry. But fantastically for plants, they're not going to go anywhere. So if you are a little bit unsure for a particular type of plant, exactly what feature or features that you need to photograph, well, the good thing is that it's going to sit there and be very patient while you take photos of it. And so you can really take your time and make sure you capture all the necessary characters. So really, so again, like the insects, I'll delve into a few specific examples. But if you encounter a plant and you have no idea what it is and you have no idea what features to capture, there's really just a nice standardized set of 7 or 8 photographs that you can take. And no matter what the plant is, probably 85 to 90% plus of the time, you should be able to get an identification. So the first photo, and one of the most important, and I think one of the most underrated, and this is often a photograph that is missing from many citizen science records, is a photograph of the whole plant. Because this type of photograph actually includes many other types of metadata as well. So it includes things like the soil type, the habitat type, the height of the plant in relation to other things nearby, what other species are growing, whether the plant is erect or prostrate or creeping, and also perhaps most importantly out of all of these, it tells you the growth habit of the plant. So is it a shrub? Is it a tree? Is it a creeper? Is it a herb? And all of these pieces of metadata and information that I just mentioned can all be really, really useful for identification. Then you want to get photographs of the leaves, and it's really important here to not only get photos of the top of the leaves, but also the underneath, and I'll explain in a second why this is important. A photo of a flower, if it's present both from above and below, and again, I'll explain why this is really crucial. Photographs of fruit if they're present, and if it's a kind of a weird looking fruit, it's often useful to take multiple angles. Photos of bark. Now, if you've got a tree, it's useful to get both the main stem or trunk as well as individual branches. And then finally, just a photo of an entire branch or branchlet itself, just so you can see the arrangement of the leaves and the fruit and flowers on that branch. And overall, when you combine these 7 or 8 photographs, as I said, more often than not, you're going to get a reliable species identification. If we deal with a specific example, the family Asteraceae. So these are the daisies, a super diverse family with tens of thousands of species across the world, many, many species in Australia as well, both native and non-native species. Most of the time, or maybe not most, but a lot of the time, unfortunately, when I see people who might be very, very new to citizen science or natural history uploading photographs of Asteraceae, it's often a single photograph from the flower directly above, which sometimes is very much identifiable. But in other cases, this actually doesn't allow you to get an identification to species. So just 2 examples here of Senecio and Brachyscome. There are many, many species in these 2 genera, and often you've got multiple species that look either very similar or in fact almost identical when you photograph them from directly above like this. So if you do encounter some kind of daisy, whether it's a native or not, one of the most important photographs I can actually recommend is taking a photo of that flower head from underneath, because here this allows you to capture the Filleries or the involucral bracts. So these are all these individual little skinny structures that you see. And you can see in this really nice example, I've got 4 photographs, all from the same genus, but 4 different species. And you can see how radically different the Filleries are in each of these examples. Now, diving into a more specific example, I thought this would be a good one to include just because it's such a familiar genus to so many people, particularly if you've ever been involved in bush regen. So if you were to see this flower head out in the bush, if I was to show you a photo like here, I'm sure that many of you would be able to immediately say, yes, I recognize that plant. It's mother of millions. It's a really, really noxious weed, quite a bad weed in Australia. But what if I told you there are actually two different entities that have a flower head that looks exactly like this? And it's actually the leaves that you need to differentiate between the two. So not only is mother of millions, so that's the one on the left-hand side here, quite a bad weed and quite a widespread weed in Australia, but you also have this second entity, a hybrid, Bryophyllum hoftoni, for which the flowers look absolutely identical. But when you look at the leaves, you can immediately see there is a very obvious difference between the two. And so this is actually a classic example of one where, although when many people encounter plants and they're photographing them, the flowers, of course, tend to be the most salient feature for most species. And so they tend to be the thing that gets either photographed the most or even photographed exclusively. This is a great example of a case where if you ignored the flowers entirely and only took photographs of the leaves, you would still be able to get a identification of the species. So just a really nice example of, yes, flowers are really important to photograph in almost all cases, but you also need to photograph those other features as well. And just to round things out here with another classic leaf example, for anyone who's ever been out in the rainforest, they'll know that most of the time flowers, fruits, all those seemingly useful ID features are often 30, 40, 50 meters up in the canopy. And so when you are actually taking photographs of rainforest plants, most of the time what you're photographing is the leaves. And many rainforest species have these really cool structures called domatia underneath, where there are these little pockets or hollows. You find them at the junction of the main rib and the secondary veins of the leaf. Things like mites and ants are often in them, and they're really important. And the number, the presence or absence, the shape, the size and color of these domatia are often really important identification features in these rainforest species. So if you do find yourself out in the rainforest and you're photographing leaves, make sure you get photos of both above and then flip them over and get underneath as well. And with these tips, hopefully you'll be well on your way to documenting as many rainforest species as you possibly can. So I might swap over to Jasmin here. I could spend hours and hours talking about all the different tips here, but we will now dive into the world of fungi with Jasmin.
Thank you so much, Thomas. That was fantastic. And for anyone that has a question, please put it into the question box. We'll have some time at the end and really keen to make the most of the two experts we have here today. So with that, we'll turn to Dr. Jasmin Packer. Thank you for being here. Jasmin is passionate about discovering deeper connections among people and nature to protect our most threatened communities. And Jasmin is the Vice President and a citizen scientist for the FungiMap Project, Australia's NGO for fungi, and also Wildlife Research Lead for University of Adelaide's Bandy Bunch Research Lab. We also wanted to take this quick opportunity to congratulate Jasmin and her team as being the runners-up for the most recent Citizen Science Prize for South Australia. So with that, I will pass it over to Jasmin. Thank you so much.
Thank you, Erin, and hello, everyone. Thank you. So now that we've heard from Thomas on some great tips across such a diverse range of groups across kingdoms, we're really keen to hear from you. So we would love to do, so we're going to do a 30-second brainstorm in the chat, and we would love you to, so click on the chat and type in, we'd love to hear what you've been sharing records of, whichever platform you use, whether it's iNaturalist or Frog ID, whichever one. So what you've been sharing records of and what sort of information you include with those records. So 30 seconds, off you go. And we'll come back to this question at the end, and we can reflect back on where you are at the starting point of what you've been sharing already. But in this little session, so building on what Erin and Thomas have shared, I'm going to focus a bit more deeply into increasing the number of species that you can share, are sharing through whichever your platform of choice is. So helping you to move those pretty pictures towards more of the research grade data. That's really, really important, and that makes it up into the Atlas of Living Australia. And also helps you to learn the names of the species that you have around you. So whether it's because you like to know what things are called, or whether you're really competitive and you just want more species on your profile, we don't mind. All of it is really, really helpful for protecting biodiversity in Australia. So really keen to help you to increase those numbers in your species. And I've included Rona's there because he's amazing. I find him so inspiring. He's one of our top recorders in terms of observations and also identifications in Australia for our fungi. So if you need inspiration, check out what Rona's up to. So to help you increase those number of actual species identifications, we're going to do three things. So the first part is thinking about what is it. Second is around what's unique about it, and it is, you know, whatever it is you've just discovered, you're finding and wanting to share. And then the third and fourth is what's it living within and where. So really, I guess, building on from a different perspective, these fields which Thomas has already touched on. So thinking about them from a different perspective through fungi. And many of you would be familiar with some of our native fungi, but then there's also some other things that you might not be. And so thinking about fungi is a way to kind of think about some more of those shared approaches that Thomas has touched on. So first of all is, what is it? So as we know, as Thomas has highlighted, plants produce fruit bodies to spread their seeds. So we think of macadamia and quandong as our natives. Whereas fungi produce what we call spore bodies to spread their spores. And exactly as the plants, as Thomas has shared, those spore bodies which are producing the spores, so the reproductive structures, are really, really important. So we want to think about those because they are how fungi are classified. So we know mushrooms and truffles, but there's also other types of spore bodies. So this is our first clue. The spore bodies and the overall structure of them are our first clue to help us to classify fungi and to help you, I guess, have a name for what you're sharing photos of. So these are some of the overall structures that you can think about. So as Thomas said, you know, it could be a tree or a bush in the world of fungi. It could be a mushroom, an earth star. These are just some of the types. We'll just touch on some of the types. So a jelly, an earth ball, a bracket on a tree, hubs or corals. These are both corals.
Jasmine, I just might interrupt you. Some people are having trouble seeing your slides. We can see them most certainly, but we're not quite sure
what's happening.
What's happening?
Yeah. Thanks for jumping in, Erin. So I've clicked share and it's showing as sharing. Let me think. If I do
Maybe.
that, is that better for people?
It's really hard because we can see them. They're beautiful. Maybe stop sharing and try to share again. So sorry about this. I'm really not sure.
But one thing I don't know if you did, Jasmine, did you just share that individual window? Can you share your entire screen and then go into the presentation? I don't know if that will make a difference rather than.
Yeah, thanks. So I've stopped sharing and I'm jumping into share.
So yeah, just share your entire screen rather than just the PowerPoint.
Yeah, okay. Entire screen. Do that. And then I click on my slides.
And then just, yeah, go.
Just projection and hide.
Yeah, so I reckon that should work now.
Let me just check.
Just check in with the chat, perhaps.
Yeah.
How's that?
Well done. It's always
Mm-hmm.
some technical. Yep. Okay. We're gold.
Yep, thumbs up.
Yeah.
Everyone can see?
Thank
Great.
you.
Okay. So I've just gone back a couple of slides just to show you this one. So just that highlighting that we're talking about this first question of the first part of great species records, which is what is it? And that fungi produce spore bodies to spread their spores. And those spore bodies that they produce is what we see and is really important. And it's the first clue to classify fungi. So it's the first clue for us to be able to work out what is the name, and that will help us to move towards it being a species of observation beyond the pretty pictures. So there are different types of spore bodies, and we think about this as the overall structure. So like Thomas was talking about, is a bush or a tree. So there's different types, and these are some of the types, some of the more common types. So mushrooms, we know, but there's also earth stars, jellies or brains, earth balls, brackets on trees, clubs or corals, these are both corals, and discs or cups, these tiny little cups. The second part of working out a great species record is a close-up of defining features. So we think about the spore bodies. So it's like zooming into those spore bodies, the part of the spore body where the spores are held. So let's have a look at that. So it's what's most unique, what's the defining features? And by defining features, we mean this little checklist here. So this is some work that we've done with Murrayland Riverland Landscape Board in South Australia, and we've produced these target species, and we'll talk more about them later. But we know that in this region, it's a target species because we know that in this region, if there is a mushroom that has these features in the orange box, these defining features, it's kind of a unique cluster of characteristics, we know that it will be a ghost fungus that glows in the dark. We can identify that from photographs on iNaturalist. Some other species, we need a microscope. These we can do by photographs. So we're looking at these defining features. In a mushroom, these are some of the important features. So this spore holding surface in a mushroom is likely to be gills or pores or teeth. There can be some others, but these are the main three. And then there's some of the other features to look at as well. So as Thomas had said, you know, looking at the top of the cap, but then also underneath where the spores are held in the gills. So in some other types of fungi, the spore holding surfaces. On the left for the corals, it's the tips of the coral branches that are important. For the bracket up the top, it's the underside, there's some pores there. So you can see there's just some different examples there of different spore holding surfaces that are really important, and that when we're taking photos is really good to zoom right into so that we can see the detail. The third and fourth part of a great species record is what it's living within and where. So living within, so as we know, so fungus is mostly out of sight. So it's deep within, working away where we can't see them, and the spore bodies that are produced that pop up when it's moist are just the very tip, like an iceberg. So the fungus will be living mostly within soil, animal or plant, or a dead animal or plant. And that's what we call substrate. So we're really interested in the substrate that it's living within. And here's some examples. So the soil moss leaf litter, by that we mean living within a leaf or a little twig. A living plant, dead plant like a log, or even dead animals, so in or around bones. That's the substrate, number 3. The fourth is, where is it living? So you can think back to Thomas's photo of the shrub with the four-wheel drive in the background, and so that was in a desert landscape by the look of it. So this is within a stringy bark forest, open forest. This is just a summary slide, which is inspired by something that Thomas had found and shared from the Canadian iNaturalist group, which is a really lovely, just kind of summary. I just really like the Canadian approach of the summary of what are the key points to look for different types of organisms. So this is a summary of what we've been talking about for mushroom fungi. So again, it's just those 4 key points. So number 1 is that overall structure. Number 2 is the close-up of where the spores are held. For this, it's a mushroom, so closing up of the gills, the cap, how wide it is, the colour, the texture. Number 3, again, what it's living in the substrate. Number 4, the habitat. So just to touch on the substrate here, we've talked about not just the fact that it's in a living tree, but that it's at the base of a living stringy bark and the species, so Eucalyptus obliqua. So that's the host. And that interaction can be really important to identify. It's also really important for us to know more broadly. So again, just that summary. So these are the photos that we would love you to share in a platform like iNaturalist or the other platforms that you use. So these collection of photographs that will help to identify 2 species if it's possible. It gives us the best chance. And then if we do the same for a coral, so number 1 is the overall structure. So something that shows the whole organism. Again, as Thomas flagged. Number 2, that close-up of the tips and the branchlets. So it's important of what the end of the tips are looking like. These are like little kids' stubby fingers, and how dense, how many of the little branchlets. The substrate in soil and the habitat, again, a forest. And then what you can do is take those photos and pop them into iNaturalist. And if you choose a project like FungiMap Australia, then in that project, we prompt you to help you to remember to comment on the habitat and the substrate. And here you can see we're saying substrate or host. So if you can say stringy bark or the species, that's really helpful information. And moving on to tips and tricks to help you to collect that data while you're out there finding things that you're wanting to share and perhaps find out a bit more about, and perhaps even to protect. So the first is to use target species. So I mentioned them earlier. So these are species of fungi that you can learn, we can all learn and identify from photos without needing to use a microscope. They're also ones that we want to and need to know more about. So they've got distribution maps with black holes or a lot of white space, a lot of area that we need to get out and survey so that we can find out more about their distribution. And that's important because it helps us to understand extinction risk for them, how at risk of extinction they are, but also those others that are more cryptic, so those that are a bit more hard to survey and perhaps are not so common. So those that are threatened. So having those that are easy to find and we have lots of records helps us to understand those that are more threatened. So how to find target species of fungi? There's 2 ways. One is our FungiMap target species. We have 200. You can find them on the website. And this is a favourite shot of Sophie Green, our national coordinator, and the Fungi Down Under book, which has the first 100. But you can also find them on the website. A second is regional targets. So we've worked with, as I said, Murraylands Riverland Landscape Board. A lot of the photos here that I'm sharing are from Nicola Brown from the Murraylands Landscape Board. So thank you, big shout out to Nicola and the team. So these are 20 species. Again, there's the web address so you can jump in and have a look. They occur in lots of places in southern Australia, so you might be able to find them in your area as well, or chat with us about developing target species that are specific to your region. Second is location, and Thomas has touched on this already, so we don't need to cover it much, but just a reminder, yes, make sure your location is on upload straight away because you'll pick up if you've forgotten to put your location on, and you can adjust that. Reminder that you can, if you're worried about any species, you can always put an obscure or private on them. And this is just to show you how you can do that. So when you're uploading, you can choose that, or you can edit and go in later. And these are first species that need extra protection. And lastly, fabulous photos, of course. So we all love gorgeous photos. and one way that having beautiful photos can help is by having a hero photo as the first photo in the platform, so that as people are kind of swiping through, looking, you know, at beautiful photos and things that they're interested in, they will also spot things of like, oh, I know what that is. I can see what it is because it's a great photo that's got all the defining features on that first slide for the record. So here's an example of that. So this photo, you can see we've got a mirror just showing the gills at the bottom there. We've focused in on the gills. It's a bit blurry, but yeah, we focused in so you can see that the colour of the gills and that they are gills, the colour, and that they're quite close together. But then we can also see the cap and the stem and the markings on that stem, that kind of chestnutty colour. There is the spores that have fallen out of the cap. So there's actually a lot going on, and there's a lot of information in that one hero photo. And actually what we could do is go into edit, hold on that photo and slide it across so it becomes the very first photo for that observation. And it would just make it easier for people to identify it if they're kind of flicking through. Other things that are important, just briefly, are the scale. Again, Thomas has touched on that. So just a reminder, you can use your hand or anything else that you happen to have with you, and that's really important. And how many there are. So great to have the zooming in, but we also need to know how many there are, because that helps us to identify the species, and it also helps us to track changes over time in that location, which in terms of land management and climate change is really important. Lastly, try to capture breeding activity, whether it's, you know, juveniles of a wallaby or a kangaroo or a favourite bird that you're seeing. If you're seeing juveniles, it's really important breeding activity, and the same with fungi. So if you can capture them at the different life stages within that spore body, that can also tell us about the species. And just a quick note that of all of the photos I've shown, they've all been native fungi, apart from this one, which is pretty, but it is a weed. So lastly, so cute shot, but what is it is the big question. So think back to the beginning and what you shared. Think about what Thomas has shared with his tips, and then what we've just covered for fungi. We'd love you to chat into, yeah, just pop into the chat. What would you include? What's needed here? What would you include to help name this? So to help this go from being a cute photo to being a species observation. So we'll come back and have a look, yeah, at what everyone's popped into the chat. So what is it? What are the take-home messages for you? So finally, how to find out more. So you can subscribe to our e-news, or please share fungi photos through iNaturalist in the project FungiMap Australia. And that would be great help for fungi. It helps us to learn more about them and also to protect them, which is most important. Thank you so much.
Thanks very much, Jasmin. That was a fantastic talk, and thanks to you, Thomas, and to Erin. I'm now taking over to kind of curate any questions that people have, and we've already got a few coming in. But before we do, Jasmin, I know you can't see the chat, so I thought you'd be interested in the answer to your first questions. Plants definitely have the upper hand with the most responses, followed by frogs and insects. Importantly, fungi managed at least one, which is awesome. And I think there has to be a special shout out for nudibranchs. In terms of what people were recording, there's a really great range, everything from just a simple photo all the way through to taking weather conditions, habitat descriptions and the like. So a really fantastic array, and I think it'll be really interesting to see how people's observations improve in our citizen science platforms as a result of this presentation. In terms of the questions that we've been asked, I might throw to you first, Jasmin, and then to Thomas for the first one. So there's a question from Alice about quite a few people have voiced concern over incorrect identifications happening on iNaturalist, particularly taking them to research grade when they aren't correct. How much of an issue is this, and what can be done to address it? Jasmin, if I can throw to you first, because you've got a curated project on iNaturalist, what are your thoughts?
Yes, thanks, Cam. Great question. This is one of the reasons that we've in the past been super careful in curating data for fungi through FungiMap, and we used to have, you know, every single one being carefully looked over by experts. We found that by going to iNaturalist, we can collect a whole lot more information. It does mean that it's harder to get that level of research grade data, the proportion of research grade data. So yes, it's a challenge, but there are ways that we can address that. And so people getting to know and recording, you know, with these parameters that Thomas and I both talked about, so really clear information and photographs and across the whole suite of those characters is really important. That can help and increase confidence. The other is for fungi particularly, target species can be really helpful, and people just focusing on the target species that they know is really important. And then we do have expert mycologists, some of whom are citizen scientists, who do that background work, and obviously can, even if a species is to research grade, people can still comment on that.
Okay, thanks, Jasmin. Thomas, what are your thoughts?
Yeah, great question. I'll be pretty blunt here. I think that the perception of the error rate across research grade observations across iNaturalist is often greatly exaggerated and very overblown, and that the true number of errors is actually much significantly lower than a lot of people make it out to be. Or people take one very esoteric group of organisms, which might be very difficult to identify from photographs, and then extrapolate that error rate to all taxa, including things like birds and tropical fishes, that the error rate is probably under 1% for many records. So I think, I guess the most important point to make here is that, yes, there are some taxa for which research grade records may be incorrectly identified, but the incredible benefit that iNaturalist offers is that these records are correctable at any time by anyone, anywhere, no matter how long ago they were uploaded. Whereas if we compare this to, say, physical vouchers stored in herbaria or museums, think about how many vouchers have been sitting in a museum drawer for 50 years and have never been reassessed and were identified by one person of unknown skill level. And then if someone does want to come along and redet those records, they might have to either get loans or physically go to the museum or institution. And it's a hugely time-consuming process. Whereas in iNaturalist, I can click and search for and bring up a record that was submitted at any point ever in the platform's history and correct it with a single click of a button. So it's the incredible accessibility of these records that I think provides a huge benefit. And I think I'll just round out that answer by saying there are actually a number of papers that are coming out now, not just for Australia, but for overseas countries across a very diverse range of taxa, as well as a few papers that I'm actually working on right now. And for many of these groups, the error rate is below 10%. And in fact, then the corresponding error rate in museum or herbarium specimens is actually higher for these particular focus taxa. So as one very brief example, quickly off the top of my head, termites, there was a paper that came out a few years ago about termites on iNaturalist, and they found that for the records they assessed, the error rate in research grade records and some others was around, I think, 11%. So for a group like termites, which are often pretty tricky to identify from photographs, I think that's pretty damn good. And one final thing I'll slip in is that the only way we can improve these records is getting more expert identifiers. So if you are concerned that there are misidentifications on iNaturalist, then really the best thing you can do is hop on the platform and provide your time and expertise and curate those records, and they will only ever keep improving.
Thanks, Thomas. Though I have to say, as the ex-manager of research and collections at a museum, we're going to have fisticuffs afterwards. I don't actually agree with you, but thank you very much. So another question, I think for Jasmin this time, is FungiMap interested in lichen?
Well, yes, absolutely. So lichen is a fungi, of course. It's a fungus. It's a symbiotic relationship between two organisms, one of which is a fungus. So yes, absolutely.
Thanks, Jasmin. Oh, I've just lost access to all the questions, which isn't terribly helpful. So with birds, it can be, so I might go to Thomas for this one. With birds, it can be very hard to provide corroborating evidence to go with the sighting for citizen science. Photos are difficult to capture, calls only heard sometimes. What are your thoughts about how to improve those types of observations, Thomas?
Yeah, good question. So in this case, it may be a matter of picking the appropriate platform rather than, say, the appropriate evidence per se. So this is a great example of where eBird might be a really useful app to input those records where, you know, you're an experienced birder, you are able to recognize these birds by eye or by call without necessarily being able to capture a photograph or a sound recording. And so eBird facilitates the entry of data points without the evidence per se. Alternatively, apart from that, I guess, yeah, just carrying a camera around with you. And I know I get very frustrated when I try and track a bird for 15 minutes and it completely eludes me and I don't get a photo. I think in the case of birds, probably the sound recording is the way to go if you really do want to get that evidence. And again, the bird isn't going to be calling all the time, but especially if you're dealing with little thornbills or finches, or if you're in a dense eucalypt forest, getting that call is going to be much easier than trying to get photos. So I think those are probably the two big tips there. Start to fall back on the sound recordings where possible as probably a more reliable piece of evidence than the photos, or I guess an easier piece of evidence to capture. Or think about, well, since iNaturalist is focused on the photos and the sound recordings, if I'm not able to get those, but I am able to reliably identify these birds by sight, then using a different type of platform like eBird, which will allow you to enter those records.
Thanks, Thomas. There's actually another question here which I might take, which is how do we identify citizen science records from the ALA download? Is it by looking at the data resource? The answer to that at the moment is yes, that's probably the best way of doing it. Then I might actually toss in a question to you both, and I might go with Thomas first, noting that we've only got a few minutes left. Thomas, what do you regard as some of the more challenging taxa groups and why?
Yeah, really great question. Aside from, so obviously probably the obvious answer is ones where you need a microscope. So in the case of Jasmin's case, you've got many, and I'll let her delve into that more, so I won't steal her fungal answer, but a lot of insects where the defining characters are genitals, for example. That seems to be the case for so many invertebrate groups. Got to look at the genitals, you got to have a microscope. But I think probably my most important answer here is the groups for which there are no experts on iNaturalist. That's something I think really needs to be improved. So there's a lot of these things where people are taking photographs, they'll upload them, the organisms are perfectly identifiable from the photographs if you're a person who has the requisite knowledge for that group. And so a classic example of this was very briefly plants in the Kimberley recently. There were people that had uploaded photographs 5, 6, 7 years ago that were just sitting as plant and had never been touched. And then you had Russell Barrett come along, who is a massive expert botanist and taxonomist who's hugely experienced in the Kimberley. And he was just going through and identifying all these Kimberley plants. And many of them, it's the first ever records of these plants on iNaturalist, or the first ever photographs of these plants in the wild. And it's just because he had that knowledge and he had access to the keys and ID resources. So I think those are probably the most challenging ones, I think, at the moment, where it's not necessarily an inherent feature of those taxa themselves, but it's the fact that you need a specific expert who has knowledge to identify them. And iNaturalist doesn't necessarily have an expert in every single group. So really, I guess, yeah, emphasizing my earlier point that if you're an expert, we want you to hop on and offer as much of your time as you can spare.
And thanks for the plug for taxonomy, Thomas. And I just drawing a link to Vanessa's comment that part of the problem with museums and herbaria is that there are so few taxonomists, and Australia desperately has a need for many more of them than we currently have. Jasmin, what are your thoughts?
Now, just to add in, I think time and space is the challenge. So Thomas has talked about some spatial places like the Kimberleys, and if you look at the ALO map, you'll see there are places where we don't have many records for taxa, but particular taxa, but even more broadly. So those places, so it's remembering when we're going on holidays or we're visiting, or it's a great excuse to pick up your swag and go and visit somewhere. So places where we really have those big white spaces of unknowns. It's that, and then over time, one of my observations is at the best at the moment we've got it to sea cucumber. So it's on a remote-ish beach in South Australia. Something we've never seen. We've been going to that beach for about 20 years. So I think it was perhaps just an event, a weather event that brought them to that beach. Doesn't happen very often. There's not many people there. And so I think there's some of the challenges. So yeah, just keeping on recording, all of us, just sharing what we see in the world around us is so important.
Thanks, Jasmin. And look, we might leave it there because we've hit the end of our timeline. So please, I'd like to thank Thomas and Jasmin and Erin again for your respective wonderful talks. And I'd encourage everyone who's been watching and listening to go out and continue taking photos, recording, and attempting to improve your observations with the fantastic hints that we've been given today. I've got 2 pages of notes on how I'm going to improve mine. So thank you, everybody, and have a great day.
Yeah. Thanks, Cam. Thanks, Erin.